Good afternoon, we'll get started. Good afternoon to everyone on Zoom, we'll get started. I think we got the whole crowd in. Good afternoon and welcome to the 64th Annual Faculty Research Lecture in Basic Science. I'm Vanessa Jacoby, I'm a professor of obstetrics, gynecology, and reproductive sciences, and I'm the vice chair of the Academic Senate Committee on Research that's hosting this event. The Faculty Research Lecture began in 1957 at UCSF to recognize and celebrate the outstanding career achievements of UCSF research faculty. And for those of you on Zoom who are not witnessing this, I'm pretty sure that this is the first time since 1957 that most people in the audience are wearing matching t-shirts that have the honoree's image on the back playing an electric guitar. So just huge rounds of applause for the Jim Wells fan club here. The nominations for this award are made by UCSF colleagues who consider the scientific contributions of their peers and their mentors and submit nominations to the Academic Senate Committee on Research, which then awards the recipient. I'd like to do a big thank you to the committee members who work really hard to review the outstanding applications that we receive. We invite you to also attend the Faculty Research Lecture in Translational Science, which will be held in 2 weeks here in Byers Auditorium and on Zoom at 3:30 May 10th. And also the nominations for next year's Faculty Lecture Series is now open through June 10th. So please consider submitting a nomination to one of the 3 categories: basic science, translational science, or clinical and population science. The committee welcomes and encourages nominations of faculty from historically excluded groups or those underrepresented in medicine and science. Please submit your nominations through the Senate service portal, and if you're on Zoom in the chat, there'll be a link put there right now. Thank you, Liz Greenwood. Today, we are very proud to honor Dr. Jim Wells, a truly outstanding UCSF faculty member who has had a major impact on science at UCSF and far, far beyond. I'm really pleased to introduce his nominator, Professor Michelle Arkin, who will introduce Dr. Wells. Professor Arkin is the chair of the Pharmaceutical Chemistry Department and co-director of the Small Molecule Discovery Center at UCSF. She was a postdoc in Professor Wells' lab at Genentech and then left Genentech with Dr. Wells to help start the Sunesis Pharmaceuticals. In 2007, Dr. Arkin moved to UCSF to help Dr. Wells and Professor Adam Renslow build the Small Molecule Discovery Center. It was wonderful to read the nominating letters and support from Dr. Arkin, and I'm really excited to hear her talk about Dr. Wells today.
Thank you, Vanessa. I have known Jim for almost half my life and for almost all of my professional life. As a postdoc advisor, Jim was very patient, barely rolling his eyes at my questions like, where is the adrenal gland? And what does SAR mean? Many other things about his style were surprising, like expecting paper reviews to be positive, or at least not personal and rude, which was new in my experience. And he didn't expect you always to be working. He would help one particular graduate student with his online chess game, for example. But mostly, and I admit that I learned most of this in long retrospect, Jim was wise. He knew that students would work, that student would work for days straight when inspired and to let him follow his own rhythm. And he taught me a very important principle of mentorship, that your job is to put opportunity in the mentee's way. You can't make them pick it up. He put many opportunities in my way. Some I tripped over, some I picked up. And for both, I thank him with all my heart. We had our ups and downs starting Sunesis. On the ups, when we made a great hire or had a great Halloween party, and on the downs, when a big project turned out to be an artifact and we didn't publish it. Or when we hired badly. Oh, so badly. But we learned a lot. And eventually we both learned to leave and come to UCSF. And now I am his boss. So you see, it always works out for the best. But you aren't here because Jim was a great mentor or a pretty good underling, but because he is a world-class scientist. And if you don't already know him well, you will probably experience the following set of thoughts during his talk. What a silly name to call that new technology. What an elegant use of that technology. What a general principle of biology I've just learned. Oh, why didn't I think of that? Again and again and again. Turning a protease into a ligase, displaying functional proteins and enzymes on phage, alanine scanning mutagenesis, hotspot theory of protein-protein interactions, principles of small molecule discovery at protein interaction interfaces, reversible covalent fragment discovery, antibodies that recognize small molecule protein complexes, degranomics, surfaceomics, sniper, rescan, bioreact, and so forth. And so you are in for a treat. And now, without further ado, I turn the podium over to the 64th Academic Senate Lecturer in Basic Science, Professor Jim Wells.
Wow, that was amazing. And you have pictures I have to get because I've lost many of those pictures and I really want them back. A secret source. Wow, this is amazing. Thank you all for being here. This like my whole life is here. It's just it's amazing. Thank you all for coming, especially my family who's here in the front row. Carol, my daughter, Hope, and John and Bill, Janet. Where's Jackie is over there. Marcia, thank you for coming, all family. And then alums, great to see you in those blue shirts. I love those things. I've got one myself here that I'm going to put on just after this. So, and it's really an honor to get this award because this is an amazing place to do science, and to be recognized this way is really, oh, I can get all teary-eyed. But I want to tell you that about this title, Pirating Biology for New Inventions. So this is a place that's amazing for discovery biology. I would say that there are very few places in the world that have the kind of biologists that this place has. And actually, I actually pirate your biology. I'm interested in the mechanisms you discover, the machines you discover, and taking those machines and engineering them so that they can do new and better things, sometimes for discovery biology, but also for new therapeutics. So I want to tell you about that today, some of our work. So, oh, I got a clicker here. Okay. Sorry. Turn it on. Advance there. Okay, so my life began here, right here in Washington, D.C., when this naval aviator came back from World War II and married this, my mom. John married Joanne, and they were said to be, they met at George Washington University where they both were, and said to be the best dancers in the entire college of their class. Now that's actually a big deal because dancing then was like a major art form and a major way of interacting. So they got married. Here they are galloping down the aisle. And then they started a family. And here's my brother, John, here. You can see him with my mom. I came along. I was number 2. And we then went from Washington to Menlo Park. My dad was a physicist, and he joined then the new institute called Stanford Research Institute. And my mom became a social worker, and so they moved out to the West Coast, and And of course, John and I were then attired in our Western gear here. You can see us armed to the hilt with these 6 guns. John has some sort of long-range rifle there. And then, there's a jug of elixir that's sitting right there too. Then we were joined by 2 others, my younger brother Bill, who's here, and my sister Rachel. We actually get along really well. You know, we had our sibling stuff, but we actually do stuff together. We take trips. We're going to Europe together, a boat trip together this summer. It's really amazing. The other group that I really hung out with in high school and junior high school was this one here. One of our parents named us the Good Word. I didn't think anything of that, but then people started, are you guys a Christian band or something? I mean, I got nothing about Christianity, but we did not realize that we were proselytizing it. And anyway, so this was a great experience for me. I was with this group for 5 years, from early junior high school through high school. We played at hundreds of different gigs. Really, sometimes we were playing twice a week. I still have this guitar. I can't say so about the plaid pants though, but it was really a blast. And then after high school, I made my way to Berkeley, which is an amazing place. And I was a psych major and a biochemistry double major. I took biochemistry from the legends of biochemistry, Dan Koshland, for instance, Howard Schachman, Jack Kirsch, Bruce Ames. These are icons, and I learned about that, and I really appreciated molecular machines and what they can do. Catalysts, you can catalyze reactions with these proteins known as enzymes. But maybe the best thing about it was I met my wife, Carol, there. Here she is. She's a fantastic jeweler and an artist, super creative. And we spent a lot of time in her vehicle we called Big Bug. This thing allowed us total freedom camping wherever we went. Here we have the kitchen out there. You could just stop the thing, open it up. You have a kitchen, throw some canned goods in there. You're eating in about 5 minutes and driving on to the next place. I spent a couple gap years between Berkeley and starting in graduate school trying to figure out what to do. I was a scrub nurse for about a year. And then I decided, well, I didn't want to do that. It felt too like I was a body mechanic or something. And then I got really interested in science. And so I applied to graduate schools. And so was waiting. I wasn't actually getting a lot of bites until this guy here, Ralph Yant, who was at WSU, called me to say, hey, we really wanted me to come. Totally convincing. Ralph was Dan Koshland's first postdoc. So he had a long history of enzymology himself, working on myosin. An amazing mentor for me. I mean, I learned so much from Ralph, writing and in science. I mean, rigorous science. He was amazing. I worked on, but I'd have to say, when Carol and I came into town, there was what I would call a WTF plus moment. Now, what the, right? Plus means actually it came out okay at the end. So I've had a bunch of them that are WTF minuses. I won't be talking about those today, but I will be talking about some pluses. Because here it is, we're coming from Berkeley and we're going into a wheat desert. You know, we didn't know what that was, but it turned out just around the corner were all these amazing places. We had a great time rafting. Carol kind of got tired of this when she nearly drowned one time on one of the rafting trips, but we had a ball. And I worked with Ralph on understanding. He was the person who developed a number of famous ATP analogs. Like you probably heard of ANP. He's the guy who invented it. Also, a number of other was like another fluorescent analogs and things like that are now sort of stock and trade and people see all the time. I worked on another inhibitor of myosin, which was we affectionately called biomud because it wasn't anything like it was described to be. It turned out that it worked by oxidizing 2 thiols on the enzyme in the presence of its substrate ATP. The enzyme closed on the substrate and would trap it there for 4.5 days. So it was amazing. I actually haven't seen many other examples since. It's one of these kind of rare birds, but it was really amazing. And also amazing was it was a early paper for me in PNAS, which is amazing, contributed by Dan Koshland, you know, the person I mentioned earlier. So amazing what goes around comes around. So I was so lucky to be in Ralph's lab. I was also, another very important event happened on August 25th, 1979. Carol and I got married in our front yard. So rather than a formal church setting, we were actually in our front yard. This house, by the way, in Pullman, Washington, it cost $125 a month to rent. And it was on about a third of an acre, about 5-minute walk from the lab. And we could even rent the room downstairs to Wiley. So we even paid less. So we had this in our front yard, quite a contrast to my parents' wedding, as you can see here. And then, of course, to go away on our honeymoon, what else would we do but go away in Big Bug, here dressed up by Jack, who's a fantastic artist. He's a professor of ceramics at Chico State. And so he doctored this up. We went off into a cornfield, actually. But it was our way of doing things, and then we did have a proper honeymoon later, I should say, in Hawaii, although there was a tropical storm then too, which. Okay, I then went away to do a postdoc, and I chose George Stark at Stanford, kind of another I was from the Bay Area, so now I'm coming back, but I love this guy. He was an amazing scientist and inventor himself. He invented westerns and northern blots. He was PALA, a number of bisubstrate inhibitors. He also gene amplification. Him and Shimke were the keys to that, as well as the JAK-STAT pathway. I think people have heard that. I worked on interferon-induced gene products, and I was purifying this really interesting enzyme called 2', 5' oligoadenylate synthetase that made these little primers. But then about 18 months into this, I got two things happened to me. First is an amazing scientific discovery occurred. I had been working on proteins. As protein chemists do, you basically just take these as you get them from nature. DNA, RNA, polypeptide fold protein, you study it, you hit it with modification reagents to probe it, et cetera, as I was doing. I love doing that. Then came along this guy here, Mike Smith. And Mike, who is an amazing person, developed this method called site-directed mutagenesis. And what it allowed you to do is, using these small little synthetic DNAs, he could then change the DNA in a cell and have it encode not the normal protein, but a mutant protein. And you could put this mutation anywhere you wanted in the sequence. You were not slave to, is there a thiol there? Is there a lysine there? Oops, there's too many of them, we can't modify. This allowed you to do it site-specifically, which is amazing. Now, there were some serious gaps, though. One is, these things are very hard to come by. There's getting synthetic oligos. Now it's obviously trivial, but it was not then. And there were not good expression systems for proteins either, very few at all. So that was a big limitation. Okay, but I was convinced I wanted to use these to make mutant proteins and find out if you could make them better. That's what I wanted to do. The second thing that happened about 18 months in my postdoc was this wonderful thing. We had these two great kids, James and Hope, who's here. There they are here, having grown up a little bit more. And with that, time to get a permanent job. You have 2 more mouths to feed. We can't just run away in Big Bug, et cetera. We got to get serious here. So I was looking for a permanent job. But as I said, I was really passionate about wanting to use site-directed mutagenesis. However, realizing that I had never cloned, sequenced, expressed a protein before, mutagenized a protein, and oligos were nowhere to be found, I wasn't going to get a job doing that. So I get a call from Brian Lawless, a friend of mine from grad school. This is what goes around comes around story, because we were in grad school together. He told me about a company he was working with that had all these toys and would be really interested in pursuing some of this kind of stuff. They might be interested. That company was Genentech, an amazing place. So I went up and I interviewed with my interview team. Here they are here, Dave Estelle and Dennis Henner, who's sitting right here, actually. Genentech in those days was a bunch of postdocs and no adult supervision. Maybe it was throughout. It's not that way anymore, but it used to be that way. And so you go up there and they're just going, yeah, let's just go for it, man. This sounds great. Do it. I was joined soon after by one of my best friends, Tony Kozikov, who together we built the protein engineering department there. And And so I was a kid in a candy shop. And I really went with the intention, Dennis, to be only there 3 years as a second postdoc, but I ended up staying 16 years because it was so much fun. So that was awesome. So what was our first project to do? It was had this question: can we improve an enzyme through mutation? Simple question. So we chose this enzyme, a really good choice called subtlysin. It's a bacterial secreted protease, and it's a protein shredder. Basically, Bacillus sends this out into the milieu to shred proteins so that it allows nutrients to flow back into the bacteria. Well, I didn't know how to clone that, but Dennis knew how to clone, and so did Eugenio. They were running the Bacillus group, and actually their secret thing for them was, I could care less about this protein. All I want is the control sequences, because this thing was known to be expressed really well in Bacillus. So I thought that would be great. So I cloned the gene, came out with a really interesting sequence in front of it, proenzyme sequence. No bacterial proteases had been cloned at this point. And turns out through work that actually Dave Agar did on alpha-lytic protease, as well as in a way at Rutgers later on subtlysin, and Phil Bryan, this is a folding chaperone and an inhibitor for the enzyme. Okay, so I put this into Bacillus. I came in the next day. Eugenio said, hey, why don't you just see if it'll go in and maybe it'll express the protein. So I put it in there. I came in, and lo and behold, on a Saturday morning, it was making halos indicating it was chewing up protein in the milieu. That was a huge deal. Allowed us to now have that whole pathway, because they had oligos there, they had sequencing, we had the gene cloned, and we had this expression system worked out. So, well, what's the first target? Now, this almost by today's standards would be trivial. It was this residue here called methionine 222. It turned out that about a decade before, a group at Procter & Gamble had shown that if you treat subtlysin, it's very sensitive to oxidants. So if you treat it with oxidants, it will cause this formation of this oxidized methionine, and that then drops the activity 10x. You just put in that single oxygen atom, and it trashes activity like that. Wow, that's amazing. Why is that? Well, it turns out you don't need to be a molecular modeler to know that the active site serine is at 221. It's right next door. So it's sitting right next door. And moreover, this methionine, I should say, is conserved in all of the subtlysins that have been sequenced. So it's like, what are we going to do? It's got to be that. So we wanted to replace it because we didn't want this to happen, but we didn't know what to put in there. So me and Dave Powers, who's here, he was my first RA. He's now at Abbott running antibody engineering. He and I developed this method called cassette mutagenesis. Now, what I mean is this is using double-stranded DNA, inserting double-stranded DNA, as opposed to the single-stranded versions that Smith was using. Why would we use that? Because if you're using Smith's method, you had to have everything P32 labeled. The efficiency was super low. You're sloshing in P32. In fact, people were being found at Genentech to carrying P32 back to their cars and their carpets to their house and things like that. Yeah. In fact, one of us in the lab was named Hotfoot for having, I won't tell you who, but some know who that was. So Dave and I basically generated these 20 mutations like shooting ducks in a barrel using this sort of method. This is now people would now call, I think these days they would call this deep mutational scanning or something. But this was the first example of doing that. And so, oh, I got ahead of myself. So we had a betting pool. What's actually going to work the best? There were some on the far conservative thing. I don't know if Dennis was one of those, saying nothing's going to work. Yeah, it's a conserved residue. It's found everywhere. You're not going to find anything that works as well. Just a substitute for the methionine. Now, I was sort of optimistically a little bit in the middle, and maybe I was the most optimistic. Actually, it turned out I think I was right. There were actually some of these residues did work quite well, serine and alanine. So if you take and you put subtlysin in neat hydrogen peroxide, 1 molar hydrogen peroxide, this is the curve that happens. You lose activity, it just gets trashed within 2 minutes, it's gone. But you could substitute in these non-oxidizable residues and it was preserved, which is amazing. So that was really the first example of improving an enzyme with mutagenesis here, at least in my recollection. So we also then went around and started changing a bunch of other residues in the specificity site. And we found, wow, we could make it work, steer specificity for substrates 10 to 100-fold by making contact mutations in the binding site. That was amazing. We could also introduce disulfides into the protein very easily. In this paper, we introduced 7 different disulfides that provide structural stability to the proteins. So then you say, oh, why would you be interested in doing this? What's the goal? A guy named Jonathan McQuitty, who was in the business development group, came down the hall to say, hey, Jim, did you know that subtlysin is the largest industrial enzyme market? I mean, that's how curious I was about that. It was all about making the enzyme better. And so one thing led to another. We immediately got in touch with the Procter & Gamble people. It came out. We started a partnership. And it's now sold in Tide through a company that was spun out called Genencor. And it's not a small product after all. I just looked it up. The sales of these engineered subtylysins, and all of them found now, are engineered and expressed recombinantly using these cloned genes and mutants. $1.5 to $1.8 billion in sales. But on 2,000 tons of enzyme. In fact, the first, this represents, I'm not kidding you, this was the first truckload of subtlysin, recombinant subtlysin that was produced here. And I think it outstrips any recombinant protein to date in terms of the way it was made and the amounts that were made in that process. So it was pretty cool. Now, we also went on to make more specialized versions of subtylysins. We could do this, as I mentioned, this is a protein shredder, but we made another variant using a technology that Paul and I noticed was possible, which is something called substrate-assisted catalysis. And what this was that this is the cutting mouth of subtlysin. This is where all the shredding is happening, and this is a victim substrate that's coming in. It's going to get shredded by that serine there. These residues are really critical. In fact, Charlie Craik simultaneously was mutating another related enzyme called trypsin, and showed that if you mutate that alanine, that aspartic acid out, enzyme activity just absolutely gets trashed. So, but we saw that if an imidazole histidine comes from the substrate, it can actually dock over this. So maybe we could remove that histidine and let the enzyme use the substrate to assist in catalysis. And in fact, that worked. The enzyme activity went down overall, but it went down dramatically for anything other than histidine. And so that was actually sold by Genenase. This is the name was invented by Paul Carter. Genenase 1, sold by New England Biolabs for, I think they discontinued in 2013 for site-specific proteolysis. So that was pretty cool. There were other ones that came behind this, better ones, actually another derivative of subtlysin. The other thing we built was, and Michelle alluded to this, was a peptide ligase. So normally this enzyme is a shredder. It's tearing stuff apart. With 2 changes in the enzyme, we made it into a peptide ligase that would take an ester and connect it to an amine. And actually that turned out to be, and not hydrolyzed. It would just take esters like this and react them with an alpha amine, and you would get a ligated product. We made actually whole proteins this way. Dave Jackson did. Or Tom Cheng, who's here, made semi-synthetic constructs this way. Amy Weeks also recently engineered even the specificity of this to make it better. We made cyclic peptides. And later, as I'll show you, we use this for what we call cellular N-terminomics. Sammy Maros, who was actually a student with Charlie and Nick Agard. So, well, as things are going on, there's a little bit of competition going on at Genentech between subtlysin, which we had, and another thing that was being developed for breaking up blood clots for heart attacks, known as tissue plasminogen activator. That's now, many of you know that this is used first line for patients with heart attacks. They get it in the ambulance. So Genentech was developing a TPA, and we had subtlysin like this. So we wondered, like, okay, I didn't feel so good about this, so I bet this thing can beat up the TPA. maybe not in sales or helping people, but you know. So what we did was we had this protease battle where we took TPA, we put it in, just incubated it by itself. There's a band showing you the protein's still intact. So nothing happens when it's just incubated by itself. If you put in cetylysin by itself, nothing happens to it. But if you mix cetylysin with TPA, you can see that's 0.2 hours and it's gone. Okay, it was trashed. Well, I presented this actually at a Asilomar. Bob Swanson was there, who was the CEO of Genentech, and he kind of didn't take lightly this. So I got a call the next when we got back from Asilomar. Dave Martin, who was then director of research at Genentech, he was here as a faculty member. Maybe many of you, I see nodding heads, will remember Dave. He's a great guy. But he said, "But Jim, we're now a pharmaceutical company." And I said, "What the fuck? We did all this great engineering." So he asked, "Can you find out how our HGH works," which was their first product. "Can you make it better?" I had no idea. I had no idea. We didn't even know how this hormone worked that allowed short stature kids to grow to normal stature. And I love structure, brought up at the knee of Bob Stroud on the importance of structure. And the only structure we had was this thing here, which was a picture from a PNAS paper by a group at Monsanto of pig growth hormone. They wouldn't release the coordinates. They would just show you, "Oh, we solved the structure, here it is." Well, actually, that was a pretty useful thing. Brian Cunningham, he built actually a wire model based on this picture that turned out to be quite useful. And I'll tell you a little bit about that just in a sec. Okay, so we didn't know where things bound, but we knew there was probably action on the backside of this hormone based on other studies that we've been doing called homolog scanning. And Brian Cunningham said, "Well, look, I'm just going to mutate everything on that face of the hormone and see if we can find out, you know, what's really important for binding." And this is the first alanine scan that was done. Michelle mentioned that. Any of these bars that are above this line here cause huge disruptions in binding affinity. And we actually found some that would enhance binding affinity, which was really wild. And it turns out that when you map those onto this graphical model or this wire model, they all sit on this face of the hormone. It's about the size of my hand on this thing. That's about the patch that is, okay? I can pass this around if you want to fondle my growth hormone here. So we then, things started taking off at this point. One of the things is it was growth hormone also bound other receptors. This is a well-known phenomenon. Hormones bind usually multiple receptors. Do they use the same face? Would they use that same interface that we found for growth hormone? So we took all those mutants and we then looked at them for binding the prolactin receptor. I think Jermaine Fu, who's here, had done some of this as well. And maybe there she is. Lo and behold, we found that these same patch of mutations, they didn't bind all the same in the same way, but these two functional epitopes actually did overlap with each other. And the other thing that we found is you'll see this ZN thing there. We were getting a lot of variability in our data, and we figured out that there's actually a zinc cation that sits right smack in the middle of that interface. Now that turned out to be also interesting because we were getting, when we were looking at our growth hormone, we were finding that we'd sometimes get a dimer peak and sometimes a monomer peak for the hormone. And it turns out that the hormone itself can self-dimerize with zinc as well. So it made a zinc dimer. Turns out that growth hormone is released from your pituitary, and the highest concentration of zinc in your body is in your pituitary. And we then found that that's the reason why is because they're all the growth hormone is stored as that zinc dimer. And that turned out to be useful. That's why I have this symbol here, pirating biology, because Genentech now formulates the growth hormone in the presence of zinc because it's so much more stable. That was really cool. So we wondered, could we engineer things to bind better? Steve Bass, who's here in the audience, was a postdoc who, there he is right over there. He noticed this paper from, who would become very famous, George Smith won the Nobel Prize 2 years ago for this technology called phage display, which George had developed to find out, could we find peptides that could bind to different epitopes. The way that works is you can take a library of genes encoding peptides, put them into these phage as a large pool, and then select for a target that would bind the best phage from here, and recycle this and identify new peptides that would bind to a target of interest. That was huge. So we then wondered, well, could we get proteins to do this? So Steve, and then joined by Henry Lohman, 2 postdocs in the lab, developed what we call monovalent phage display. So we took growth hormone, that's growth hormone, that wire model there, just sort of a cartoon version of it. We put the growth hormone receptor on a bead. We then made mutations in this thing and selected for ones, asking if we could find ones that would bind better. And in fact, we could. This is a collection, all of these chicken pox here, the size of this represents the improvement in binding we got when each of those were discovered by phage display. And in fact, when they were all compiled together, we could make it bind 400 times tighter than the natural hormone. So you think nature's actually better? I mean, you can make things bind better using technologies like this. This was astonishing. And that actually inspired Steve to come in costume. Here he is as what we call a phage head here. Okay, so then about this time, a piñata broke, and that was from the work of Tony Kozikov and Bart de Vos. De Vos means fox, I'm told, in Dutch, so hence the fox here. And this was a promotional party where you're forced to drink tequila shots until you drop over. Some Genentech know about this. They solved the structure of the growth hormone receptor complex actually using protein that Germain had learned how to express really well, the ectodomain of the growth hormone receptor, and it found 2 copies of the receptor. So we've been studying just this interface over here. We really didn't know about this interface here, but we had biophysical data suggesting that there was going to be another there. So this was huge, and for 2 reasons, well, multiple reasons. This was the first member of the cytokine receptor class to be crystallized. Now, there's now 50 different cytokines. They all essentially look like this in various forms. Stoichiometries can vary or whatever. Bob Stroud solved the EPO receptor structure, and it looked very similar to the growth hormone receptor structure that Tony had solved. Of course, Tony was trained at the knee of Bob. He was his first postdoc, I believe. And so we wondered, so there's 2 things I want to tell you about this. One is it relevant for signaling? And it turned out it was relevant for signaling. Germain Fu, also over here, did some really elegant studies where we mutagenized this site 2 in growth hormone and showed those acted as antagonists. So if you don't dimerize like this, you don't drive signaling. And that turns out to be really, really useful. Why? Because there's a disease called acromegaly, which is a growth hormone excess disease, and people want to block that. The only way it's blocked now, the first thing people do is they take out your pituitary gland. You can do that once. It's a very dangerous operation, actually. And then if they don't get it all, you're going to live with growth hormone excess and get giganticism, which is a really painful way to die. I think the lifespan is like 10 to 15 years, and it's not fun. So we built a growth hormone antagonist, which basically was one of the most highly engineered proteins at the time, maybe still. It used modifications in binding site 1 to improve affinity. Remember those high affinity variants I mentioned? We use those there. And then we use one of Germain's sites to block the dimerization so that we could make this stick as a monomer to the receptor and prevent the thing from dimerizing. And now that got, through the work of Ross Clark and Chip Scarlett, a small company that they formed, they took this system, they developed it, ultimately then got it approved in 2003. And most importantly, Aetna agreed, yeah, we want to use this because it's the best treatment for acromegaly. So that was very cool. Another thing that came out of this was work that Tim Claxon had been doing with Brian Cunningham, developing, finding that now with the structure in hand, we could actually mutate everything that was in contact. We didn't have to use those funky wire models anymore. We could use a structure. And Tim and Brian, and then Ken Pierce after him, found that not everything in the interface is actually critical in binding. There's these small hotspots in the center of the epitope that are really critical. And that was really amazing and found to be general too. So in fact, here are the 4 other interfaces. They all show the same hotspot phenomenon. Why do you care about hotspots other than it exists? Well, there was a lot of interest in building small molecules that bind and block these interfaces. And there was a lot of skepticism that you could prevent binding of big molecules to them. But if there are hotspots, maybe you could do so with small molecules. Well, this is an amazing group. Michelle showed a bunch of pictures. This is one of our ski trips that became a tradition even today. My lab goes away on ski trips. And this is another amazing group at Genentech, the Postdoc Group, which I was fortunate enough to be the first senior mentor of. In fact, Joff Baker, who's here, is right there. I think Dennis Henner, who's here, is right over there. I think there are others here too that may be in this picture, but it was really an amazing group. So, but I was really getting anxious about, I really wanted to understand this question of, can we target hotspots with small molecules? And Genentech was really not going to do that, not put the chemistry muscle into it and not really go after it the way that I wanted. And also, I was really fascinated with the notion of starting a company. So with this sort of mantra in mind, and really that was it. I mean, we're just going to, and a really talented group, many from my lab. There's Michelle there. Brian Cunningham joined. Warren Delano, who many of you use Pymol. He's the inventor of Pymol. Dan Erlinson, who's right over here, and Andrew Braisted. They were the first crew. Joined also by people like Marcus Ballinger, who came a little bit later, and others. It was co-founded with John Ellman, who many know from the combinatorial chemistry days. Not everybody here was at Synesis. This is Greg Weiss. He's now a PI at University of Irvine, UC Irvine. So we, building the ship while you're sailing, it was our experience. Yo ho ho ho, pirate's life for all of us now. So there were a lot of bumps in the road. The first one started off right here. How would we pay for the first month's rent? There were some bumps in the financing. It didn't happen. And yet there was a lease that we had on this property, and finding property was really, lab space at that time was very tough. So either I was going to accept that we're going to be 3 months down the road to try to find a place, or I ended up putting down a deposit with my credit card. That didn't go over well with Carol, I'd say, but so a WTF plus moment. But the funding did come in. Now, this place wasn't the highest rent district. In fact, this is our next door neighbor. There were a lot of pit bulls over there and Doberman pinchers we'd hear at night as we were going to our cars. So that the basis for Synesis was really about fragment-based discovery, which was pioneered by Stephen Fesick, who's a brilliant drug discovery scientist at Abbott at that time. And the idea is that normally you take big molecules and screen them and then have to chisel them around to put a square peg in a round hole. But Steve came up with this really clever idea of identifying fragments that you could then assemble into the binding site. So that was really cool. So can we target hotspots with fragments? The disadvantage of this method that Fesick had was that these things were non-covalent. They bound very weak. You couldn't get structures of any of it. So we developed, I should say, Dan Erlinson, who's here, and Andrew Braisted, who passed away. Remember the Braisted lecture? That's named after this fellow, amazing guy. We invented this technology called tethering for site-directed drug discovery. So what you can do is you can put thiles around a binding site you want to find things in, take this library of compounds, allow it to interact, and by thiol disulfide interchange, if something actually sticks in this site here, like this red circle, then it'll get tethered into the protein, and we can detect that by mass spectrometry. So we started then this amazing project. Actually, this was led by Michelle and Andrew, Brian Raimondo, and helped also by crystallography by Chris Thanos, who's actually here. And what was done was to introduced thiols all over this receptor binding site. We didn't have the structure at that time, but we knew it bound up in this region. And these are like fishing docks. And so by fishing from each of those docks, we found compounds that would bind in each of these positions, but they weren't all the same. It's like if you're fishing from one bank, you catch trout, from another bank, you catch salmon, from another one, you might catch sharks. And you can start to then assemble them, taking, for instance, this piece here, putting on a biaryl piece, generated an intermediate compound, and then with a library that was built over this northwest area, we found a molecule that was 60 nanomolar that bound to IL-2. Freestanding compound, this is the structure of it, and that was an absolute breakthrough in the protein-protein interface area. Nothing had been found like that, which was really amazing. And the other thing that was really amazing, it taught us how adaptive these interfaces are. For example, if you just take the wild type IL-2 studied by crystallography, you'd see that this molecule wouldn't actually dock into it. That site isn't actually there. And so by solving these intermediate structures, we could see the actual morphing and docking of this interface. So when the first fragment went in, there's the wild type. You know, you get this little dimple here where that molecule bound, and then this opening of this channel, and then this reorganization of this whole area here when this carboxyfuran piece binds. So I'll let this go through, but you can see that this part over here does not become apparent until the rest of the molecule is there. So at the time, there was a raging debate about induced fit versus conformational trapping. This is a beautiful example of conformational trapping because these sites are not evident until you build the rest of the molecule, which can then find it. These interfaces are very dynamic, so they actually open up and they're breathing all the time. You can't see it in this way, but think of it like it's sort of shivering. Okay, so Kinesis was growing as a result of that, and other things that we were doing. We had a high throughput screening library that was great, and finding things. We had 5 partnerships. So we went from this crew here to then our own building and this crew here, which was really amazing. We also had an amazing portfolio. We had a number of kinase inhibitors. We had these. We actually had 3 industrial collaborations that were supporting half that group, and we had advanced things from hit ID all the way into clinical trials. So at this point, there was another transition for me, which was, I should say also, one of these became a drug through 2 colleagues at Genentech then, John Bernier and Tom Gaddick. They started this company called Sarcode. They licensed this molecule. They developed it. It's now a drug sold by Novartis, and I think it's about a $500 million drug. So that was pretty cool. But I was, once you get into development, it's a bit like watching grass grow. Things just go very slow and methodical. All the resources are there. And I really wanted to get back to doing invention. So I wanted to be untethered to plunder to heart's content. So that's when I joined UCSF, which is an amazing place, really. I am amazed we get paid for the UCSF people here. I think I get all hands up where we are amazed we get paid for what we do. I know it's a pain, we complain about it, but hey, it's better than flipping burgers. And one of the people that was really instrumental in my recruiting was Bob Stroud, who was on our SAB, and we'd been talking for months about this transition. And then really stepped up when I was talking with the guy who actually had the FTE and the money, Tom James, who was chair of the department, and Ron Vail, because I have an appointment in both Pharm Chem and CMP. And this woman here, Marianne Coda-Kimball, who's absolute delight person and super supportive of basic science in a school of pharmacy, which you would think is not going to be where you'd expect the greatest sort of basic science. But 2/3 of the departments are here, and it's an amazing group. I also got funding to begin to really get small molecule discovery going here. With first hired Adam Renslow to run the chemistry group, and then Michelle convinced her to come over from Kinesis to run the biology group. They then took over this department. Now Michelle runs it entirely. She's director of the SNDC. And they both have thriving labs with students and postdocs. They're doing great. So that was a real success. I also joined these two amazing departments I mentioned, pharmaceutical chemistry and CMP, the real leaders in science of drug discovery. I would say, I don't know, maybe I'm getting too boastful here. I don't think there's a better, Kayvon, maybe you can tell me, is there a better place for doing chemical biology? You hear? Yeah, no way. This is the best place on the planet to do chemical biology. So things are going along okay. And a year and a half in, Tom comes to me and tells me, hey, Jim, how about you be chair? I'm going to retire. So that wasn't what I sort of signed up to do. It was kind of, but it was actually a WTF plus because we got to build and hire a bunch of new people. And it was, I did it for 7 to 8 years. And it was actually, I really enjoyed it. I wanted to get back to doing just research. And we were fortunate enough to then have Matt Jacobson take over as chair. And just recently, Michelle Arkin take over as chair. And she's right. She is my boss. Maybe it was always that way, Michelle. I don't know. But so I'm going to tell you in the last 15 minutes or so, or I guess I have 20 minutes, which is good timing, 4 parts of our research at UCSF, triple AS, as I call it. The first one was allostery. So we had been looking for protein-protein interfaces using tethering, right? We could put things there and try to assemble them. But by some serendipity, we started to learn that you could actually use it to poke and prod for allosteric sites. Allosteric sites are sites distant from the active site, but if you poke them, you can affect the enzyme. It's like, think about it this way. If you're eating your dinner and then someone comes along and tickles you, you're not going to be able to eat very well, right? You're going to be laughing, you're going to be doing something. So at a distance, you can affect function, and that's what allostery does. That's the best analogy I could think of at the moment. Others will think of better ones. This was the founding pirate ship. Let's see, Justin Shear is here, Dennis Wolin, Sami Maruz, Hikari Yoshihara. All of these 4 were at Sunesis. They all came over with me. I was so lucky that they did because they helped build the lab group. And then they were joined by Jun and Deb Dada, my first student. Ironically, Sammy was at UCSF. He was in Charlie's lab, and Hikari was with Tom Scanlon. So here, when I told them about going back to UCSF, it was quite a little bit more of a conversation. You know, hey, I just was there. I'm coming back. Okay, so that was what goes around comes around. But what we found, I want to show you this example. This was actually discovered first at Sunesis and then expanded much more at UCSF. This is the enzyme called Caspase. It's actually a molecular demolition expert that drives apoptosis. I'll tell you a lot more about it. It has 2 active sites, one here and one there. And right in the middle there, we wanted to find inhibitors for this enzyme for blocking inflammation because we wanted to shut the enzyme off. By tethering, we found some inhibitors here that turn the enzyme off. So way away from the active site, an allosteric example. And so I'm showing you now a cutaway of this here. This is like a quadrant of it there, but now just in stick view and ribbons. This is where, and so when a tethered compound that we found binds in this interface, it causes this dramatic rearrangement to occur. Okay, it goes from on, now it's off, and then it'll go, if that thing leaves, it then goes back on. And in fact, this is an example of induced fit, like Koshland had proposed, because there really are 2 states in this, and this molecule stabilizes the off state. Now, the way it goes off is that the substrate normally sits like right here. That's the active site cysteine right there. And when this binds, it thrusts this arginine out. There's no way the substrate's going to work. So that was really amazing, totally fortuitous. So WTF plus. And it's present in almost all other caspases have this site in it. We showed it, Justin Shear, who's here, showed that this is present in caspase 1, his work here. We also showed it was present in caspase 7, also caspase 5, and found allosteric inhibitors. So that's pretty amazing that we just poke and prod with tethering, we can find these things. So that then unleashed a bunch of projects, both here at UCSF and some legacy from Synesis. Caspases, as I mentioned our work, but Adam and Michelle have some beautiful work on Caspase 6, showing another allosteric site. PTP1B, which was studied extensively at Synesis, been further studied in Jamie Fraser's lab in a collaboration with fragment-based discovery. C5a receptor with Henry Bourne, the KICS with Anna Mapp's group. She found tethering hits that bound in a core that stabilized it, allowed her to get an NMR study. Probably the most famous was this work that Kayvon's lab did, taking the tethering library with John Ostrom and showing they could find a tethered compound that would put this switch 2 region up. And now, how many molecules are there in the clinic for RAS? Is it 8? 10? And one has already been approved. And they stemmed from these early kind of discoveries. And another one that I won't have time to tell you about, but was really elegant, was work on PDK1. Jack Sadowski and Justin Rittenmeyer found an allosteric site where you could generate both activators and inhibitors. So you could either turn it on more or you could turn it off more using these sites, all based on site-directed drug discovery. So that was really cool. Okay, the next A, remember AAAS. So the second A is apoptosis, because we're working on caspases. They are cellular demolition experts. We wanted to find out how they work. If you look at cells treated with chemotherapeutics, for instance, they will apoptose because the chemotherapeutic is a poison and it will cause it to apoptose. So I'll show you here. This is a picture from Bruce Albert's lab book, and you can see they just blow up. It's like popcorn popping. They actually generate, the cell doesn't actually lyse, it's generating these membrane blebs and whatnot. And we reasoned that these caspase enzymes are going after specific proteins in the cell. That was already known that they would do that, but we wondered how extensive they were, kind of like destroying this stadium over here, which I was delighted to see. This was the Dallas Cowboys stadium. And so, yeah, so we were really thrilled to see that thing go down, because they used to torment our 49ers. I wanted to show you it again, but the point is when that was destroyed, there were demolition experts that went in there and put specific charges on the struts of that building and brought it down that way. So caspases, are they also going after the quote struts of life? So you can see those flashes, you can see it there. It's an amazing thing. Okay, so we wanted to engineer proteins to study cell death. I'm going to show you, just scroll through a few of them here. This is called the sniper. This allows us to cut individual proteins in a cell to see, is that a strut or not? This is something called a netallator that Min Shuang developed and Zach Hill developed after her to look for E3 ligases in the cell. This is a collaboration I had with Gray Davis and the student who worked between us, Ashley Smart, who developed this light-activated caspase. So it has this love domain, which if you flash light on it, it unfolds. And so we put it in there in a way so that it's the caspase is taught as a proenzyme, and then it's cleaved and it's released. So we put that love domain in there, and you flash light on it, it suddenly gets active. So we could put this into cells and neurons and find out what individual neurons groups are doing based on shining light on flies that Gray was talking about. But the sort of the granddaddy of them for us was this enzyme, again, subtle ligase for N-terminomics. Because this enzyme was key to us finding out broadly, what are the substrates for caspases and a number of other proteases. You can see the number of papers that we've had here. And the way that this works is that if a protease cuts a protein, you generate a new alpha amine, this NH2 group here. And subtle ligase loves these and will put this ester onto the alpha amine of that cleaved product. And it's got a biotin on it, so you can pull it out. So we could absolutely then pull out all of the substrates before and after we induce apoptosis. And so this group, the stars of N-terminomics, started with Sammy and then Nick and then a whole bunch of other folks joined in, including Arun Wita, who did a whole scan of omics work, RNA, DNA, looking at what happens during apoptosis when you activate caspases or even identifying new biomarkers for them. So we learned a lot from this work. For instance, we identified 2,000 different aspartate cleaved substrates. 1,400 of them were new to the literature. So about 10% of the proteome can be cleaved. It cleaves not only after aspartate, which it was named after, but it actually can cleave after either phosphoserine or glutamate. It cleaves only once or twice per protein. It often goes after protein-protein complexes, so kind of cooperatively. And these are the kind of favorite foods that it loves to attack very rapidly. mRNA, microRNA, and mRNA processing proteins, kinases, cytoskeleton. These are the things that it just absolutely attacks first when it's bringing down the cell, which is really interesting. If you want to find out, is my protein cut or not, you can go to our website, Degrabase, and look up, put in your protein accession number, and that database will tell you whether or not we observed it or not. Okay, the next A is antibodies, which is now becoming a huge part of what we do. Antibodies are critical reagents for research in medicine. They are probably the biologist's closest friends because these are used so extensively for biodetection, FACS, IP, Westerns, PET, proximity labeling, CyTOF, NGS. But most of these commercial reagents are actually not so good. None of them are renewable. Many of them are not renewable. They're made in animals, so you lose them after the batch is made. So you don't get back the same reagent each time. So that's a limitation. But antibodies are also amazing drugs. They can be built into a whole bunch of different formats to attack tumors. And these days, and going up to 2026, they will represent over half of pharma revenues will be this class of proteins. Antibodies are. So it's an amazing thing. So in talking, sort of bringing back the band, my buddies from Genentech, Tony, remember, he was director of our department. He went off to University of Chicago. Dev went off to University of Toronto. He was a postdoc in the lab and had taken my position when I left. He was doing great there, but they had developed some new libraries that could be used for phage display libraries based on some of the pioneering work of Greg Winter, who won the Nobel Prize for some of that work, identifying antibodies that could bind to targets of interest, shown here in these hexagons. So we said, wow, maybe we could use this for building renewable reagents, because each of these is a genetically defined molecule, permanent. You can express it. And these scaffolds expressing E. coli, so you don't even have to do it in mammalian cells, which is a lot more difficult. So we built this machine to enable this, because all of these things, I can remember Dennis talking to you about, shouldn't we build a robot that would do phage display? Because all the steps are routine, and they over and over, you're just washing, binding. So enter Mike Hornsby, who's here in the audience, and Tet Matsuguchi, who was a grad student with, Liz Blackburn, thank you. Yes, they built the first generation of the antibot, and then augmented by Kevin Leung, who now runs the Antibiome Group here, fantastic scientist. So this is a picture of the antibot functioning. Can you hear what we wanted? We wanted it all, as Kuhn would say. So we use that actually, and other versions of it, to then generate antibodies to, or tried to over 800 different proteins, transcription factors and epigenetic factors with Tony and Dev. And using this, we actually generated about 3,000 different binders, and we were successful in generating specific binders to almost half of this group, just using this high throughput thing. So not everything at all, but to many, we got a very high hit rate. 200 of these antibodies are now licensed to Abtec, which is the antibody engineering or the recombinant antibody company. So that was cool. Now, we also wanted to build antibodies that were highly selective. It turns out that this is a protein, a model protein here. After they're made, they don't just sit there, they're actually modified. They're modified with a whole bunch of what are called post-translational modifications. Some of them shown here. A dominant one is phosphorylation. And these aren't just ornaments on a tree just to decorate it because you want to put something out there. These control the enzyme. So they're put on there to control biology. And we wondered, could we make antibodies, recombinant antibodies that would be specific, for instance, to phosphorylation? And so this postdoc then, JT Korber, with Bill DeGrotto, who's here. Bill was then doing a sabbatical in the lab. They developed this new scaffold for, what? Actually, well, okay. I think you're on the patent too, Bill. Maybe we should take you on. Well, no, Bill was amazing. And so we built these antibodies good for phosphoserine and threonine. Kurt Mu joined and he built an antibody, or actually cloned an antibody and improved it for phosphotyrosine. And Xin Zhou with Lei Wang in a collaboration, Xin's now at Harvard, she built ones that were very specific for proteins, folded proteins in their phosphorylation sites. In addition to phosphobinders, this group here, led by Xian Lim and Jia Zhou, they really were a team in this effort. We could generate antibodies that were specific to single proteolytic events in proteins. So that's amazing. When you know, the only thing that happens is you nick the protein, that's it. Very subtle change. Using differential selection, these two were able to generate antibodies that were totally selective for this proteolytic event. That proteolytic event occurs prominently in pancreatic cancer, and this target is on its own probably too toxic to attack with just a full-length antibody. But they showed by attacking the cleave form, it's much safer. So adding, when things are upregulated and expressed, and have an additional thing on top of it, you can actually get greater selectivity. Now, we also were interested in conformations that proteins undergo. I showed you some in the caspases, for example. And this group here, Junjun Gao with Dev while he was at Genentech, and then joined later by Nathan Thompson, a postdoc, were able to make antibodies that were specific for different conformers of caspases. So that showed that we could actually lock conformations with antibodies. Very subtle differences. You haven't changed the composition of the molecule. All you've done is just change the state it's in. So that was cool. Quickly then, as I start getting toward the end, I want to tell you about another project we used antibodies for. This being to take advantage of this phenomenon known as, or discovery by the famed Stuart Schreiber and Jerry Crabtree of conditional induced dimerization. This is a very useful tool for being able to turn proteins on and off in a cell with these engineered dimerizers. But it's all based on rapamycin. We wanted to expand that palette to go beyond that. And so these two, Zach Hill and Alex Martinko, developed what we call ABSIDs for antibody induced, conditional induced dimerizers, and where they could take a drug target, add the drug to it, then use phage display to identify an antibody that would sit over it, and use this in place of the switch so we could get rid of rapamycin. That led to, when joined with Aaron Simmons, his company Soteria, and Hai Tran, who's also here doing work there, working there to develop new bioswitches for T-cell engagers. So that's a really important class modality. This, we hope, will be something that actually really propels the field, because now you can tune it up and tune it down with a small molecule. The other thing we pirated from biology was in this field known as PROTACs. This is a really important new therapeutic modality that was pioneered by Craig Cruz and then Jay Bradner, where you can take a bilobal molecule like this. One binds an E3 ligase, the other binds a protein of interest, a drug target that you want to get rid of. This technology that allows you to ubiquitinate the victim protein, and this gets degraded by the proteasome. So that was amazing. It's a totally new way of getting rid or affecting drug targets as PROTACs. So we wanted to expand this to extracellular proteins because all of those examples essentially for PROTACs are intracellular molecules. So we wondered, could we go on the outside of the cell, and joined here by Adam Cotton, who was a grad student co-mentored by Ian Seipel, along with Joseph Gromsbacher, who's here, and Yui Nguyen, came up with this idea of what we call ABTACs. So the way ABTACs work is that there's a family of what's now become known over the last 8 years or so, a whole family that has they're transmembrane E3 ligase. So they have part of their nose is sticking outside the cell, and antibodies can only engage things on the outside of the cell. So we reasoned maybe we could get an antibody that would bind this and then recruit that to a protein of interest and then allow for ubiquitin transfer and then degradation of the protein of interest. So it turns out this works. It's a amazing technology, actually, and we've shown that it works for degrading a number of disease-causing proteins like PD-L1, EGFR, CECP1, and others. So very excited about that. Okay, well then, as things were going, things got, we were suddenly just stopped. COVID came along, and basically all of our labs got shut down, except those wanting to work on COVID-related stuff. So we're a protein engineering group, and this group said, all right, we're going to start working on COVID. And so we got, everyone had to come in totally masked up. We had to work in shifts. But we generated a bunch of, using all of our tools in the toolkit. For instance, we built these small domain binders that could bind to the RBD of the virus. Cole Bracken had built a VH library that would allow this, and we could combine them to make tighter binders. Also, Ashish Mangalik and his work showed this was also possible using a nanobody library that they had. We built bispecific antibodies, one that had its nose in the binding site, and another was, which we thought would be useless because this would not block the virus. But if you put them together, this bispecific VH binder, Xian Lim, Katapants, and Joseph generated these. And then also we generated these binders that bind to the they bind to the RBD by pirating the receptor that the virus is using. So it uses H2 to get into a cell. We lopped off H2, made this FC fusion, and then with Tanya Kortem's group and this husband-wife team of Adam Glasgow and Jeff Glasgow, you know, they were hanging out, you know, after COVID, and they're around each other. Oh, why don't we do this? So that's what they did, and actually this binds really well, and it actually, unlike, so since the new variants have come out, the Omicron variant, that's knocked out 7 of the 8 approved antibodies have been knocked out by Omicron and other variants. So this, however, you know, the virus has to go through H2, and in fact, Sonia Ramesh in the Antibody Group has shown that Omicron can't resist this. You know, it's using the same thing to get through. So it's a really cool binder. Then we also developed these new antibody sensors. This is the work of Susanna Elledge and Xin Zhou. Using a split luciferase, we can sample people who've been immunized and tell them what their antibody titers are. Within 20 minutes, we can get a quantitative read of this. In fact, some here have had, I think, raise your hand if you've been splucked. You've tried. See, I mean, okay, most of them have blue shirts on. But this, we can actually quantitatively tell you what your antibody titers are. I think this could be very important as the vaccines wear off, and we want to know what titers we need to protect ourselves. In fact, this is now in licensing discussions to set up a company with some people that want to develop this. Also, we're in licensing discussions around the ACE2 traps as well. All right, but our main focus here is actually to understand how cell surfaces change in health and disease. So this is the surfacome work that we've been doing. This is a huge project in the lab, has been for the last 5 years, where, or more, 6 or 7 years, where we compare normal cells and transformed cells to identify what is different on the surface of a transformed cell relative to a normal cell. And this is, you can see, we use mass spec to identify these different proteins. This was pioneered by Alex Martinko, and then Kevin came in, did a ton of this. And now this is joined by, I would say, 10, 15 others in the lab have had various forays in surfaceomics. We have different technologies. We can look at what's up in expression. We can also look at what's modified on the cell surface, like glycosylation. Using subtle ligase, Amy Weeks and now Katie Schaefer and Irene Ryu have shown that you can look for changes in proteolysis. Remember, I told you that these things are orthogonal to each other, so you can combine these things to get better selectivity. We also have a project we call the Cell Portal Project that Zi Yao is working on in the lab, where you can look at the inside proteome through display of MHC complexes. And more recently, we have proximity labeling technology going on, looking at what's in the neighborhood. So what we've done is to build a platform where we can do target discovery using those technologies I told you about, recombinant antibody selection using those methods that I told you, and then characterizing these antibodies and even putting them into animals and then advancing them into hopefully clinical trials. So that's what this group does here. Tons of projects going on. These are all of them. I wish I had time to talk about them all, but it's an amazing pipeline, and we hope great things will happen. So I'm inspired by these 3 images that are coming along here. I love gaps, terrifying gaps that look like you can't get over them, can't get through them. Nobody would be crazy enough to go over them, but then get a running start, do a pirouette, try to jump to the other side. And then after you've done that, then build a pathway that lets others by as well. So I have a lot of people to acknowledge my current lab here, which is, I'm so proud to be part of you guys. We check our badges in at the door, right? I mean, I get told all the time, that's a crazy freaking idea. Don't do that. I want to acknowledge these mateys that keep the ship afloat. In the first hatch of our lab, especially Marja Tar, who's my sister-in-law, and who can't be here, and Mary Betlak, who worked for years with Bob Stroud, was actually the lab manager for Herb Boyer here. She was also at Synesis. And now by Hazuko Olsen, she keeps, I'd like to give her a mini round of applause if we could for organizing a lot of this. Minwi Mahara, who is our lab manager, and Maria and Ula, who keep everything running. That's an amazing group. So I also want to acknowledge the Pirates. This is the current lab members. You can see them here. Postdocs and grad students. So I'd like to applaud to them. Okay. This is the amazing Alumni Group. You see that? Ending with Dave Powers, who's here. And this I want to show is all authors on all the papers that I've been on. And in yellow are all the UCSF faculty. So look at that. I mean, there's like, I counted them up. I think there's 30 faculty that we've had collaborations with. So if anybody, you hear this all the time, UCSF is a collaborative group. Jesus, this is a perfect example of it. You know, we're so lucky to be together this way. Okay. I now want to thank, this is the group that keeps me afloat. This is my family from a great gathering that we had here. Carol, my son James and his family's wife Molly, Hope over here. We have my brothers who are here. Janet is there. Where's Jackie? She's there. They're all here, which is awesome. And then lastly, I want to, of course, thank my soulmate. Ah, thank you. Hey, can I get a picture of you guys? Hold on. Let me get it in a movie form. I got to do this. Okay, there we are. All right, thank you so much. So.
Wow, thank you so much. That was inspirational. Your enthusiasm is infectious, and you can hear a pin drop in this room. And this is a group who's heard about some of these things and they were still transfixed. So thank you so much.
Yeah.
We just have a small recognition for you from the Academic Senate to add to your many awards, but this is in recognition of Jim Wells for his outstanding achievements in the basic science. And thank you so much for giving the faculty research lecture in basic science.
Thank you so much, Vanessa.
Yeah.
All right, great.
And you have some time. I'm sure there's
Good,
questions.
great.
So
I'm
go
happy
ahead.
to answer any
And
questions.
also on Zoom, I think we have some questions. So Liz, if there are questions on the chat, maybe I think we can hear you, but let's start in the room and go ahead. You can moderate yourself
Okay.
there.
Yeah. Is that Liz Greenwood? Okay. Hi, Liz. Did you have a question? Oh, maybe
Hi.
you can start here in the room. Questions. John?
So Jim, when did you first realize that DNA was in your DNA?
That's my brother here. Well, actually, it happened, I think, I really was inspired as an inventor by our next door neighbor, Mr. Van Wart, who was an amateur inventor. He had this garage, and to a 5-year-old, he'd show me around his garage and what's happening there, new mouse traps and toasters and vacuum cleaners and things. I thought, oh man, that's amazing. It was like Christopher Lloyd in Back to the Future. Yeah. Oh, I don't know if Liz had a question. Did you have a question, Liz?
There's a question from the Zoom.
Oh, okay.
Yep.
We can't hear you.
I am not sure why.
Is your mic on?
Yes.
It says. Okay.
Give it 1 sec.
Is it working now?
Yeah.
Oh yeah, there you go. Turn it
Okay.
up a little bit. Yeah.
I am up as far as I personally can go.
Okay.
So I don't know if there's something on that end, but the question is fascinating talk. I'm wondering whether you are looking into possibilities for using DNA or RNA to instruct cells to make therapeutic antibodies or other peptide or protein therapeutics. And that's a question from someone in the Zoom.
Oh yeah, that's a great question. I mean, it's kind of like the Moderna thing, right? You could put in mRNA like vaccines and get proteins out the other direction. Yeah, that company is doing quite well, obviously, on the vaccine side. And actually, before they ever developed that vaccine, they were trying to develop EPO and some of the super potent cytokine receptors. So it's definitely a viable kind of approach. Any other? Oh, question in the back.
Jim,
Oh, it's Jamie.
what's
Good
up?
heart. Oh, dude.
You're so good at naming things. What's your favorite of all the names that you've come up with?
I don't know. They're like your kids. You know what I mean? What are you going to say? I don't know.
I have a fun. Oh, good.
So, well, Jim, of all the biology you pirated, is there anything that you wish you had done but kind of missed or didn't have the bandwidth to do that or something that if you had done it again, you would have gone there?
What about CRISPR? Missed that boat. Yeah. Thank you for that.
We have another. Oh. Great. Another Zoom question maybe is someone says, amazing talk. What's your secret to building long-lasting relationships with alumni and collaborators?
That's a great question and something that I really value because that is what makes it all worthwhile, is being able to work with people, being able to just brainstorm, and we check our badges in at the door. So no one is considered, higher or lower than the other in that regard for ideas. And I think it's really important that I'm very proud of the fact that everyone has gone through my lab. I think that's an unbroken streak, has gone on to a next job that's been better than the job they had. But maybe I treat them so bad, so it always happens. But it's not like that, actually. So promoting people and hiring people that are better than you, that's a key too. So yeah. Yeah, Jamie?
I have the benefit to get talked all the time, but for the younger scientists in the room that don't have that privilege, looking back on your excellent career, what would you say is one piece of advice that you would give us as we look forward into what we try to do?
Yeah. Yeah, the question is so what advice would I give junior investigators or postdocs, graduate students, as they go through their career? I actually have a slide with a bunch of that stuff on it, but let me just pick out a couple of ones that stick out to me. I think it's really important to move on. Don't get stuck. If things are things are going to be tough, we know what science is like. It's a lot of ups and downs. I showed you a bunch of successes, but for each one of these, there's 3 that's on the trash heap. So that's really important. Resilience, having fun is a really important part. I think this has to be an enjoyable. So I feel you enjoy the ones you're with. That's a really important feature. And then no fear. Just go for your passion and what you think you can get, and just don't give up. Charlie. 3 years, yeah. That's always a question. It's much easier to see backwards than in front. The surfaceome stuff, is I'm super excited about that. Our lab is going to be focused on the cell surface, how we can find new targets out there, how we can modulate them, how we can build biologics to attack them in different ways. I mentioned the ABTAC technology. That's what I'm super passionate of, working with cancer biologists like Alan Ashworth here to find new targets to go after, building antibodies to attack them. Yeah.
Another Zoom question. Going back to Substrate, do you have any ideas why methionine 221 is conserved? Is there a biological role for oxidation to inactivate Substrate in some contexts?
That is a fantastic question. So I started the thing with MET 222, and we wondered like crazy, why would an enzyme build itself to be so sensitive to oxidation? It's going to have to live on the outside world. Maybe there is a natural sort of clock that's ticking. And in fact, that's a wonderful question. It was the same question was asked by the famed Max Perutz. Many will know Max Perutz, one of the seminal people in structural biologists. When I was at a meeting with him, he was moderating, and I was going, oh, I don't know. When I got back from that meeting, we were studying. We found that Bacillus not only secretes substrate, but it secretes a catalase. So catalases take away these oxidants. So that was amazing. There's air cover for substrate, So it's just going to put the best thing it can find there. Methionine is the best. It's not that much better than serine, but for evolution, it makes a difference. So that's what I think is happening, actually.
Well, I hate to stop the questions, but we're going to end here. I wanted to just say, I guess, just another huge thank you to Jim Wells and recognition of all that he's done. And I believe we're supposed to announce that the party continues.
Yeah, upstairs on the second floor, right? So if you just go up the stairway, to the second floor and then make a left, and then it's going to be in that that area there. Everybody's welcome, and I hope everyone can go. It would really be great to see you there. I'd also like to ask the people in blue shirts. We want to get a picture of you guys up here. These are the alums and family. I just wanted to get a picture of you guys. And yeah.
Great. And just also a big thank you to Michelle Arkin for nominating Jim Wells and allowing us the pleasure of hearing him. And thank you to the Academic Senate for hosting this. And we will see you in 2 weeks for the Translational Science faculty lecture. Thank you all for coming, and enjoy the party with Jim Wells.
Great. Thank you. Thank you so much.
Yeah, congratulations. That was fantastic.
Hey, this is really
And I know.